Hello everyone, welcome back to the recall session of INICET part 2. So in this we'll be discussing the questions that have been asked in the afternoon. So in the morning session the questions were moderate, not that tough and not that simple even, and only compared to like previous INICET there were less molecular biology based questions, but in June-July session INICET there was plenty of molecular biology based questions, right? Hardly one question was there in the morning session, molecular biology, that too cystic fibrosis, that delete mutation related question. So to begin with, afternoon session, the question, they have asked with assertion and reason. Here the assertion they have given, phenylketonuria characterized by mousy odor. Yes, the assertion is correct. The reason, tyrosine and norepinephrine are not formed. Yes, in phenylketonuria, okay, what happens? There is no metabolism of phenylalanine at all. So then how come tyrosine will be formed? How come norepinephrine will be formed? So both assertion and reasons are correct. So we will see the options now. So both assertion and reason are not true, and reason is the correct explanation for the assertion. No, both are right, but reason is also not correct. So second option, both assertion and reason are true. Yes, and reason is not correct explanation for assertion. Yes, exactly. So here the reason supposed to be why the mousy odor as phenylalanine, okay, not metabolized to tyrosine, it will take divert route to form phenyl pyruvate, phenyl lactate and phenyl acetate. Because of this phenyl acetate only mousy odor. This was supposed to be the reason, but not the tyrosine and norepinephrine not formed, right? Here the thing is both assertion reason are true, and reason is not the correct explanation for the assertion. Yes, very good. And assertion is true but reason is false. Ruled out option. Assertion is false but reason is true. This is also ruled out option. So option B is correct. And coming to second question, Zellweger syndrome is caused by defect of which following organelle? So remember peroxisomes. Peroxisomes. So this Zellweger syndrome, otherwise known as cerebellar hepatorenal syndrome, otherwise peroxisomal biogenesis disorder or disorder. So this is also defect of genes which will required for normal structure and functioning of peroxisomes. So there will be defect of the genes which are helping in the structure and functioning of peroxisomes. That's why there will be no formation of peroxisomes or malfunctioned peroxisomes, right? So that is the reason. So Zellweger syndrome always related to peroxisomes. So the correct answer is peroxisomes. So third question, long-term glycemic control in person with hemolysis is assessed by estimating fructosamine. Yes, long-term glycemic control in person with hemolysis, understand this keyword hemolysis, okay, and glycemic control. So here glycemic control in our body assessed by fasting blood glucose level. Yes, apart from that we have long-term glycemic control, that is HbA1c, right, glycated hemoglobin. But here the person is what to say, person undergoing hemolysis. So that means what do you mean by hemolysis? Rapid turnover of RBC. The RBC in quick form, they are undergoing destruction, more damage of hemoglobin, right? So how come it will be a glycemic marker or glycemic control for longer term? So it cannot be. So in that condition, alternative glycemic control, that is fructosamine. So what they have asked, fructosamine is what? Whether it is a GAG, glycosaminoglycan, whether it is a urea, or whether it is a fructose, or whether it is a protein. So remember, whenever there is a high concentration of glucose in the circulation, they started glycating what? They started glycating proteins. Okay, remember that. Okay, extra glucose will obviously start glycating the biomolecules such as proteins. Here HbA1c we are talking, right? Hemoglobin itself a protein, right? So that's why it is getting glycated. So as here, because of the condition hemolysis, HbA1c cannot be used, so fructosamine. So fructosamine is a sort of albumin. Okay, fructose is a part like a sort of albumin. So that's why it is getting glycated. So the glycated form of albumin is known as fructosamine. So it is a protein. So rest of the options are wrong. It is not a glycosaminoglycan, it is not urea, and it is not a fructose. Remember, it is a protein. So which of the following tissues are dependent on insulin for glucose uptake? So this is a question related to glucose transporters. So if you see glucose transporters, GLUT1, 2, 3, 4, 5, 6, 7, up to 7 are there, right? So GLUT1 is for brain, GLUT2 is for intestine, RBC, pancreas, and GLUT3 is for what? GLUT3 is again like muscles, so GLUT4 is for muscles, and GLUT5 is for fructose, right? So like that, and GLUT6, GLUT7 again is for like intestine, right? So here main thing is like in kidney, okay, GLUT3. So here what are the options they have given? Pancreas, muscles, adipose tissue, and brain. So pancreas is GLUT2. So we have learned that, GLUT4 muscles, which is under the influence of insulin, right? And this GLUT4 location, it is not only present in muscles. Okay, what are the skeletal muscles, cardiac muscles, and adipose tissue, right? So all these things under control of insulin. When insulin is there, then only this GLUT4 transporter will be active, glucose uptake will be happening, otherwise no. So here the correct answer is muscle, okay. And adipose tissue is also they have given, right? Brain is a rule out option. I can say is a muscle, and adipose tissue also the better answer. Two options. So the questions that have been answers have been posted by students, the two options they have given. So muscles and adipose tissue both are correct. So you have to rule out what is the extra option they have given in place of adipose tissue or in place of muscles, right? Otherwise under insulin, this both adipose tissue and muscles, especially skeletal or cardiac muscles, okay, that are under influence of insulin. Next, Hooch tragedy is related to which of the following alcohol? Hooch is nothing but, what is that? Spurious alcohol. Okay, it is spurious. What do you mean by spurious alcohol? Methylated alcohol. Okay, this is methylated. So what happens in this? So this methanol, what happened? When you consume this methylated alcohol, so this methanol metabolized to formaldehyde, and this formaldehyde forms formic acid, and this is the reason behind lot of effects, right? First it will damage the optic nerve and causes blindness, and next is it inhibits electron transport chain. So no aerobic respiration. So what happens? No aerobic respiration. So histotoxic hypoxia. Right, histotoxic hypoxia, as it is inhibiting electron transport chain. So which tragedy is related to which of the following? Here different alcohols they are given. The correct answer is methyl alcohol. Rest of the options are wrong. Sixth question, which of the following is true? To break AT bonds, what do you mean by AT bonds? Adenine and thymine bonds which are present in the DNA requires high Tm. Tm means melting temperature. Okay, Tm means melting temperature required. So to break GC bonds, low Tm required. To break AT bonds, low Tm required. Fourth option, AT bonds and GC bonds need same Tm. So remember here how these adenine, thymine, guanine, cytosine are bonded together in DNA. Okay, the thing is adenine always binds to thymine by 2 hydrogen bonds, and whereas guanine always binds to cytosine by 3 hydrogen bonds. So now here number of hydrogen bonds vary between these two. So here 2 hydrogen bonds, here it is 3 hydrogen bonds. So which is require more energy to break it, more melting temperature to break it? This one require more Tm, and whereas adenine, thymine require less Tm, right? So where is the correct answer? To break AT bonds we require low Tm. So that is the correct answer. So rest of the answers are wrong. Seventh question, identify the type of enzyme inhibition in the image showed. So here what it says, this is a Lean-Weaver-Brook plot or double reciprocal plot. So this is the what is the use of this? In Michaelis-Menten's plot, what it is very difficult to use to find out maximum velocity of the enzyme by providing high concentration of substrate, right? So it is not possible all the time. So that's why for our convenience we have used this Lean-Weaver-Brook plot or double reciprocal plot. With a little amount of substrate concentration itself, we can find the maximum velocity of the enzyme. So here what they have asked, identify the type of enzyme inhibition, right? So we have already learned there are different types of inhibitions are there: competitive, uncompetitive, non-competitive, right? And then suicidal, like reversible, irreversible. So suicidal is irreversible. So rest of the three are reversible inhibition examples. So we have learned that in competitive inhibition, Km value will be increased, Vmax unchanged, right? In uncompetitive inhibition, what happened? Both what has happened? Both Vmax and Km unchanged. In non-competitive, here you see Vmax has been decreased, whereas Km unchanged. So here this is the Km. You see here both are meeting at the same point. So Km has not changed, but Vmax has been changed. It has been come down. So in that condition, that is a example for non-competitive inhibition. That is example of non-competitive inhibition. And that's all about the questions they have posted. And rest of the few students were saying that there was a question has been asked about iron overload, then how to estimate iron overload. So liver, what to say, there was like liver iron concentration method, because iron is going to store in the liver. So liver iron concentration method, one of the method is there. And enhancers, regarding this was the molecular biology based questions, they are saying that characteristics of enhancers they have asked, right? So one of the multiple questions, I don't know what they have asked, so only I got like they have asked questions on enhancer like that. So we'll see any further, like once the full paper of INICET out, then we'll discuss in detail further with all the questions. So according to me, both morning and afternoon session questions were moderate, they are not that tough. So guys, whatever, I mean, if you have done like 70 to 80% of the syllabus of the biochemistry, then that means make it that you have done very good in exam, what I can consider, because simple questions, logic based questions they have asked. Only one question in the morning session they asked related to the cholesterol synthesis, like remembering whole pathway, how testosterone will be synthesized from cholesterol with the sequence of enzymes. It is very difficult keeping so many pathways in biochemistry. So don't worry, So one or two questions like experimental questions will be there like that. So otherwise according to me the questions were moderate. So we will see anyhow if the people who have not done well, I mean like with this you can make out like how, I mean like how you have done and where you are standing. So it's okay, there is always a better chance. So who have done well in the sense, my congratulations, and who have not done well in the sense, we will see there is always another option or other chance. So NEET PG is coming nearby, so we will concentrate on that and we will give our best. Thanks for watching.